Uh, I'm currently inviting our next speaker, Kanat Yermekbayev. And actually, I met him 8 years ago at the conference of Plangin in Kazakhstan. At the time, he was going to London to do his PhD, right? It was Norwich. Yeah. And then now he's already leading his own group. And he's head of the molecular genetics and genomics. And he will be presenting something single seat to QTL and beyond, and Zhetysu. Looking forward to your talk.
Thanks, Vanna, for the nice introduction. So, of course, it's very difficult talking after the person who only publishes in Nature and then now started publishing in Science, but I'll try my best. So we are quite excited what we are doing with, and we think that we will add some values to the real breeding practices locally in Kazakhstan and beyond the country. So title just states from single seed to QTL and beyond. First of all, I'd like to thank organizers for the nice invitation and being considered toward me. As I said, Barna said, I'm Kanat, and now I'm leading a lab which is called Genetics and Genomics at the Zhetysu University, which is located in Talkhorvan city, not far from Almaty, which is the biggest and the previous capital in Kazakhstan. And the university is called Zhetysu University, and there is a small institute called Institute of Biotechnology and Ecology that consisted of 4 laboratories. And the reason why I went this small university, they are quite ambitious, and they promised a lot of funding, of course, and we are not as rich as King Abdullah University, but they provided enough funding to set up my lab, and eventually they built a new building for the institute. So this is the background of this talk. The main motivation of this talk is my PhD research, which I did at John Innes Center under the supervision of Simon Griffiths, who received a prestigious award this year for his excellent work on Watkins collection. I'll be talking about a little bit about that Watkins collection in my second talk. And the main, this is the main outcome of my PhD research. So the main outcome is we identified and mapped the 2 height controlling QTL in Central Asian wheat. So this is just a background information, as we have some junior scientists here. So the plant breeding is a kind of art or science, selectively mating 2 contrasting genotypes that has a contrasting phenotypes in order to create a mapping or kind of segregating population. And why we do? Because this is global food security. We need more food. Of course, then we also want that food to be long-lasting and looking beautiful. And also we need our crops to be resistant to abiotic and biotic stresses. So simple scheme, selection of parents and crossing, generating hybrids, testing and releasing new variety. This takes up to 14 and sometimes 18 years, which we call kind of traditional breeding. And within that traditional breeding, you just select only for goods, just eliminating the bads. And at the end, you end up with the good, adaptable, and high-yielding genotype. However, in our current understanding, good variety is a combination of good alleles. At most important genes such as VRN, PPD, RHT, or any other genes that confirm the genotype to be resistant to abiotic and biotic stresses. In order to have this nice combination of good alleles, only traditional breeding is not enough. Unfortunately, in our region, I think including the Central Asia, all the developing countries, we cannot do some kind of sophisticated genetic analyses, and all the breeding processes based on traditional breeding. So this slide shows a roadmap uh, to do a kind of modern plant breeding. In order to identify those nice alleles, good alleles, we do need we need to generate a kind of rich diversity, and that there are several ways. As Brenda said, there is a EMS method to generate the diversity, or the diversity panel, which um, we inherited from our ancestors, the old cultivars and the land races, or any other interesting genotypes. Also, we can create kind of multi-parent advanced generation intercross, which we call magic populations, or nested association mapping populations, that comes from the several inbred recombinant inbred lines. When you have this rich genetic diversity, you will have that the phenotypic and the genetic data. At the end, you just integrate those great deal of genetic and phenotypic data, and uh, obtain a signal in the DNA that is associated with the adaptation and its components. Despite Kazakhstan being a major, not giant, but major wheat provider globally, specifically regionally in Central Asia, as I said before, all our breeding processes are based on mainly traditional breeding schemes. This is also worrying if we consider the geopolitical shifts taking place in the world, like conflict between Russia and Ukraine. It poses a great threat on our global food security. So that's why I read one paper, but I couldn't find that one. So we have to take advantage of developing countries which has a agricultural capacity like Kazakhstan in order to secure our global food security. Because of the methodologies we are using, as you can see from the graph, an average wheat yield in Kazakhstan just stagnated since the beginning of the modern plant breeding, which started in 1940s and 50s. Of course, the environment is harsh. Maybe the most very continental, because we have severe winters, which doesn't allow us to grow winter wheats, and we have also soil erosion on top of that. Shortest growing period, perhaps the shortest across the world. We have to sow and harvest wheat within 100 and 105 days. If you just delay for 5-10 days, you will not be able to harvest your wheat. And the most important environmental constraint is drought, coupled with extended heat. Coming back to the point that we need good alleles, combination of good alleles. This is a nice example. Yesterday, Johan just touched about the Green Revolution, and that this is the result of the Green Revolution. The European countries mostly using those RHT genes, they doubled, sometimes tripled the yield. However, seems we just missed that opportunity out here. Even Australia and Canada, their breeding efforts focus on collecting those good alleles in combination. Why I'm talking about Australia and Canada? Because they are very similar in environment. They have very similar environmental conditions like Kazakhstan. Even they have shifted to shorter varieties, of course, in combination of these most important alleles like PPD, VRN, and the other RHT genes. This is a paper saying although we haven't changed, there was no change in plant stature. There was a yield increase to around 0.6% per year in last century, 100 years. Although there is no there is no correlation between yield and the plant height, which we again approved in our paper using that mapping population. The same authors hypothesized at the end of this paper saying that further yield achievement should be achieved by reducing or changing the plant stature. So what we have done, of course, we crossed the Pamitiaziva, which is the variety with Russian origin, but well adapted in Kazakhstan and registered as a commercial variety. We crossed that variety with Paragon because in order to have access to those alleles, we do need a cross genotypes that are genetically a little bit far. And we created a mapping population, recombinant inbred lines, and we identified several QTL that are associated with yield, adaptation, and with their components. But it doesn't mean that we can use those genomic regions directly into our breeding scheme. First, we have to validate. Among these QTL, the plant height controlling QTL on with 5A and 6A chromosome was a kind of source for my PhD. Why? Because they have a impressive, very large additive effect. Each QTL adds to the real plant height about 9 and 10 centimeters. During my PhD with the well-established near isogenic development protocol in Simon Griffith's group, we created a near isogenic lines for those two QTL. As you can see, the difference between the near isogenic lines were significantly, statistically significant in a controlled environment. Of course, next stage is testing the effect in real field conditions. And we conducted a large-scale field trials in the UK and the main wheat growing areas in Kazakhstan, such as north and south. In the north, we grow mainly spring wheat. In the south, we grow mainly winter wheat. But however, the main wheat growing area is north, about 11 million hectares. So something interesting happened. So in the UK and the Kazakhstan 6A increased plant height. However, 5A QTL silenced in Kazakhstan were it bred for. However, it's always stable in controlled environment, also in the UK's environment. Then that was the question. Then we addressed later. Now I'm showing the the yield results. Of course, we do have those alleles, height increasing alleles. The question was why we are keeping them. When I talk to breeders, they don't. No one could answer me why we are keeping those alleles, because they don't do molecular analysis and that they don't know what genes are controlling their plant height. As you can see, large-scale field trial across the years in Alm, which means in south, Pet, 3 years in the north, Kazakhstan, Petropavlovsk, we didn't see any yield advantage coming from these yield height increasing alleles. Instead, there was a single case in Alm, ALM 2021, the yield of the 5A plus, which means the plant carrying that height increasing allele, yield was significantly lower than the short. Of course, then we thought perhaps they do need to be together in order to show their full potential. And using QTL parameting method, we collected those two alleles in Paragon's background, which is the UK variety, as I said before. And as you can see, plant height of the Paragon increased by 20 centimeters, exactly as expected, 10 centimeter from each gene, each QTL. So just for the sake of understanding, these near isogenic lines, they are like Apple phones, small tweak makes them very different, but at the same time, very identical. And that they are like twins, genetically very similar, up to 90%, but they have a huge difference in phenotype, in this case, plant height. So by the way, I'm not advertising any Apple product. It's just for the sake of understanding. So then when we generated this, we called them double nils because they carry both height increasing alleles. We tested them in the UK. As you can see on the left, the Paragon's plant height was almost as tall as the double nil, Pamitiaziva, sorry. So about 1 meter 20 centimeters in the UK environment. As expected, yield was negatively correlated with plant height in the UK environment. In Kazakhstan, in the north and the south, we tested them as well, double nails. As expected, the double nail was shorter by around 8 and 10 centimeter compared to the Pamitiaziva, which means even though they are combined, 5A QTL is still silenced. And again, we did find that there is no clear correlation between yield and plant height. Another interesting fact we found, the Paragon, even though it was almost tall as Pamitiaziva in the UK environment, it didn't lodge. Comparatively, Pamitiaziva in the UK environment, it has lodged around 35%. So it could be a nice source for the local breeders in order to increase the lodging resistance, because lodging is still be the main problem in North Kazakhstan, which is the main wheat growing area. Of course, any PhD student wants to find the gene. It's not easy, as Branda said. We didn't use, the deep and sophisticated genomic bioinformatics analysis, but we were able to squeeze the region significantly from the initial QTL region. For example, 6A QTL, when we found it was 530 megabase, which consisted of around 2,500 genes. This is due to perhaps the limited recombination rate in 6A QTL of wheat, which was reported by many papers. And we resequenced the parental genotypes, and also sequenced the exons in order to develop a kind of co-segregating diagnostic markers to generate a recombinance. So then we tested those recombinants in the UK environment and controlled environment. Through integrating them, we were able to squeeze the region from 530 to 28 megabase region, which is still significant. We have done same thing for the 5A QTL. Initially, it was 37 megabase, which consisted of around 500 genes. Now we squeeze the region to 19, but still have a lot of genes, 170 genes. Then we were able further to squeeze the region to 12.5, then 1.2 megabase, using the diversity panels, which I'll be talking in my next coming slides. So using in silico available data, genomic data, even from these 180 genes, 21 were somehow related to plant height. There is a Caubin and Zhetysu. Caubin means Central Asian wheat breeding panel, which consisted of around 540 genes, which I'll be talking in my second slide afternoon talk. Zhetysu is the Western countries' wheat breeding panel, also around 500 lines. We used our diagnostic markers and try to squeeze the region. As you can see, we get some HTB, which means the haplotype blocks or haplotype groups. Based on that haplotype groups and single marker effect, we were able to squeeze that region into 12, as I said before. Then further, we were able to squeeze that region 1.2 megabase. Why? How? Because we combined the data from 6A and the 5A, because we thought, okay, this could be, the effect could be due to 5A in case of 6A or vice versa. When we combined, we found kind of possible recombination breakpoints. The plus means poles, minus means shorts, blue means the height increasing area, coral means short allele area. So then there was a haplotype block 11. Although it contained a coral bed, it was still tall. When we look at these population structure, it consisted of very old varieties in Zhetysu which were released in 1940s and 50s. Same thing was done for 6A QTL. Interestingly, we expected Calvin to be fully fixed for this blue bit because this is the Central Asian panel. However, we expected Jediflux being like segregating like 5A QTL. However, we only found 2 haplotype blocks in both cases. Now it could be due to that limited recombination rate in 6A. But it doesn't mean that European wheat still tall, because they integressed other alleles that keeps their varieties short. Within that squeezed region, we have done some kind of in silico functional gene analysis. There are some promising genes, for example, for 6A, RLCK85, which is highly expressed in peduncle length. In wheat, in rice, sorry, the overexpression of these gene in rice shortened the peduncle length of rice. Why I'm saying this? Because during my PhD, we also very interested where these genes that drive the plant height is expressed in plant's organ. And we mapped the effect of 6A QTL into internode 1 and internode 3. The effect of 5A was mapped to internode 2 with overlapping effect on internode 2. So this is very coincides with that hypothesis. For example, for 5A, AXR1 gene is expressed in peduncle 2. So this is the map of all genes that wheat 6A contains and wheat 6A and the 5A contains. If you look at the 6A chromosome, most of the plant height controlling genes identified in wheat located on 6A. Our gene on top. So we really expect this gene to be a allelic version of RHD25 or RHD25 itself. However, 5A QTL could be a new gene because on 5A is only 12 RHD12 is available, and the location of our gene is far from the RHD12. So this is a conclusion, which I may just read it down. The tallness of Central Asian wheat is controlled by 2 major QTLs, which have large additive effects. The production of nils showed us that the height increasing alleles confer no yield benefit, and therefore it could be a interesting genetically, also practically breeding back, which means decreasing the height of the Pamitia ziva with the short alleles coming from UK wheat paragon. So with this, I'd like to thank my supervisory team at that time, and for your attention. Thank you very much. I'm happy to take any questions.
Yeah, thank you, Kenneth. It's really interesting. Are there any questions? Solomon?
Kanat, thank you very much for your interesting presentation, your work. So my question is uh, belonging to the mapping populations. Just you use the biparental mapping population, yeah? What do you think about which mapping populations are very suitable for quantitative genetics? For example, yes, we know that multiparental is good, but maybe which one? NAM or a magic population or natural populations? What do you think?
Yeah, of course, if you have magic population or non-population, there is a great resolution compared to the reals because you get a kind of large mosaic genome compared to recombinant inbred lines. But in our case, the task was very simple to identify the genetic regions for plant height. So most people use recombinant inbred lines for many purposes. And when you compare with double haploids, the recombination rate in reals is higher than the double haploids. And the problem with the recombinant inbred lines was the generation time, which could take the up to 8 years if you don't have any controlled environment. But this problem is now solved by speed breeding, as Brett mentioned.
My next question, you said that there is no correlation between plant height and yield, yeah? But how I know a lot of references highlighted that there is a correlation between the yield and the plant height. How can you?
Sorry, you mean plant height being correlated positively with yield when it is tall or short? For example, in Europe, it's negatively correlated. When it's shorter, there is an optimum plant height. Of course, it shouldn't be too short and too tall. And they found that this is an optimum plant height, and this plant height is the very good plant height for good yield. In our case, mostly we keep very tall varieties like landrace like varieties for feeding purposes. They say we grow tall wheats because we have old combines.
I mean, how the plant height is scored, and then the grain number, grain size might be increased.
Sorry, Azad, can you repeat the question?
So how the plant height is short, the yield might be high.
Oh, okay. So as I said, during Green Revolution, just scientists identified RHT genes, and that they are kind of insensitive to exogenous applications like gibberellin. When you just put the fertilizers, when you use that alleles and put the fertilizer, it doesn't grow tall. What it does, they directly through all nutrients to the spike rather than other plant organs like the stem and the leaves. Did I answer your question?
Thank you. Good.
Yeah, thanks for this very interesting talk. You might be aware, working in this topic, that plant height is also associated with the length of the coleoptile. And there is a certain limit, and it was observed that this shortest cultivars may have a problem under stress conditions where a longer coleoptile is required. do you consider that in your collection, in your selection? So I actually would always have a look at that with all accessions. And as far as I remember, this correlation is not that strong in genes which do not directly interfere with the gibberellic acid biosynthesis and perception. And you showed one candidate gene interfering with BUG1, which is kind of interacting with BRI1. And I would consider that one of the most important, one most interesting.
It's a very interesting candidate genes. We are just keeping there. There is a list that contains a lot of genes, but I'm just showing them as a most promising candidates. If you could offer any ways of these cloning these genes, I'll be happy to chat with you after or anytime.
Thank you.